Hello, this is Yashvin. In this video, I'll be solving paper 2 variant 2 October November 2020 9700 biology. Question 1. Figure 1.1 is a diagram drawn from a photomicrograph of a transfer section through part of a leaf. The arrows in figure 1.1 show the movement of water through the cells of the leaf after it has left the xylem vessel. So water moves in the leaf through symplast and apoplast pathway. Symplast pathway includes the movement between cells crossing through plasmodesmata without cell walls, whereas apoplast pathway would be crossing cell walls without entering the cells. A water from xylem can enter cell A and then move to cells B and C without crossing their cell walls. So that would be through symplast pathway. The cell structures through which water passes from cell A to B are not visible in figure 1.1. Name the cell structures through which water passes from cell A to B without crossing their cell walls. The structures would be plasmodesmata. To explain what causes water to move from cell B to cell C. Movement from cell B to cell C occurs due to the difference in water potential between these cells. As the farther away a cell is from the vascular bundle, the lower the water potential. 3. Name the pathway taken by water cells between cell A and cell C. So that would be simplest pathway since they do not cross cell walls. B. Most of the water that arrives at the leaf passes to external atmosphere. With reference to figure 1.1, describe and explain the sequence of events occurring between point P and point Q. Between point P and Q, you're supposed to outline and describe the events that occur during transpiration and explain why they happen. So water vapor, water travels from cell A and then eventually to cell P as well, and then enters the cell wall of cell P. Because the water potential in the airspace would be lower than water potential in cell wall of cell P, which is a spongy mesophyll cell, water vapor would evaporate from its cell wall into the airspace and then it would eventually diffuse out of Q, which is stomata, into the air down the gradient, because the water potential would be higher in the airspace compared to the air outside. Whenever the question is related to transpiration, a few points that you must always mention in your point is that the cell wall of spongy mesophyll cells provide large surface area for evaporation of water vapor. And it's essential that you mention water vapor, cell wall, and mesophyll cell. See the actual diameter of cell D in figure 1.1 along the length xy is 25 micrometers. Calculate the magnification of the image and write down the formula used to make your calculation and show your working. So you're supposed to measure this length xy in millimeters or centimeters and convert it to micrometers, which should be somewhere around 36 to 39 millimeters according to the mark scheme. And then you would multiply that with 1000 to convert to micrometers. I'd be using 36 millimeters over here. The formula for magnification is image size over the actual size. And I'd convert 36 millimeters to micrometers, so that would be 36,000 micrometers divided by 25, which gives the answer as 1440 times magnified. 2. The treatment for people with active tuberculosis lasts 6 months and involves a combination of antibiotics. This is usually very effective if the person has a susceptible strain of Mycobacterium tuberculosis, the causative agent of TB. Table 2.1 summarizes one recommended treatment strategy that involves a combination of antibiotics. A. Susceptible strains of Mycobacterium tuberculosis will be killed using any of the antibiotics listed in table 2.1. However, combination treatment is preferred as it is one method that can be used to reduce the impact to society of antibiotic resistance. With reference to table 2.1, explain how combination treatment for TB can help to reduce the impact of antibiotic resistance compared to single antibiotic treatment. using a combination for longer intervals of time, for example 6 months, ensures that all of the bacteria are killed and none of them remain to develop resistance or transfer them eventually to other bacteria as well. And how that happens is, if even if the bacteria are resistant to say rifampicin, they would be destroyed by the rest of the antibiotics. The chances of those bacteria remaining and forming a reservoir of infection would be reduced by using a combination. And then to complete your answer, you can explain that by using the mode of action of different antibiotics. So I'll be using in my answer, I'd be using rifampicin and isoniazid. Rifampicin binds tightly to an RNA polymerase molecule close to its active site. This affects the activity of the enzyme. Okay, so initially the question mentioned that rifampicin enters bacterial cells and inhibits protein synthesis, and now we're told that it binds to RNA polymerase, but not to the active site, but somewhere close to it. So we can make out that this antibiotic affects some event during the mRNA transcription, since that is where RNA polymerase is required. This affects the activity of the enzyme. Okay. B1 RNA polymerase catalyzes the formation of messenger RNA from DNA. State the term for this process, which is transcription. 2. During the formation of RNA, a number of events occur that involve the action of RNA polymerase. Suggest ways in which rifampicin could affect the activity of RNA polymerase. So RNA builds phosphodiester bonds between RNA nucleotides, and how the form is, the RNA nucleotides bind to the active site in the enzyme. We need to recall what we learned of the enzyme topic. We learned that enzyme activity can be inhibited by two types of inhibitions, competitive and non-competitive. And since this antibiotic binds to a site other than the active site, maybe it's acting as a non-competitive enzyme inhibitor. So it does not allow the enzyme substrate complex formation between RNA polymerase and its nucleotides, which means it does not allow the formation of phosphodiester bonds between nucleotides. Because it does not allow binding between the substrate and the enzyme, it may have impacted the shape of the active site. cRNA polymerase is composed of 5 different polypeptides. Gene RPOB codes for one of these polypeptides, known as beta subunit. One or more mutations in a specific region of RPOB results in strains of Mycobacterium tuberculosis that are resistant to rifampicin. In these strains, mutations often occur in two DNA triplets in this region in position 526 and 531. Table 2.2 summarizes the results of an investigation into 7 rifampicin-resistant strains A to G that have amino acid changes from position 526 and 531. The table includes change in mRNA codon from position 526 or 531, amino acid change that has occurred as a result of mutation. The minimum concentration of rifampicin required to inhibit growth of the bacterial strain, MIC, the number of other mutations occurring within the specific region of RPOB. 1. Complete table 2.2 to show the amino acid changes that have occurred in strains F and G. Okay, so in F, we have the new codon UUC. Let's code for UUC if it's present anywhere else in the table. So it's here, and it codes for the amino acid PHE. So that would be the answer here. And UAC is also present in the table and codes for TYR. 2. With reference to table 2.2, list the strains of Mycobacterium tuberculosis that show the greatest resistance to rifampicin, or in other words, the strains which require the greatest amount of antibiotic to inhibit growth of bacterial strain. Okay, now observing the table, you'd see that A requires less than 50 micrograms per centimeter cube, which is 2 less. And we have B, which requires greater than 100, D, which also requires greater than 100. F and G also require greater than 100 microgram per centimeter cube of antibiotic, which is the minimum amount. So these four are going to be the most resistant strains. Three suggest reasons to explain why strains C, D, and E show resistance to rifampicin, and why they show different levels for resistances to rifampicin. Now you need to think back to the mode of action of rifampicin to be able to explain why they have resistance. So rifampicin binds to a site other than the active site on RNA polymerase in bacteria, and that means in bacteria that are which are resistant to rifampicin, the antibiotic is not able to bind to the enzyme RNA polymerase, or in other words, the shape of the enzyme has changed, and that change is due to the mutations involved in the gene which codes for beta subunit, which is the gene RPOB. And as for different levels of resistances, because resistance is caused due to mutations in the first place, different levels of resistances may also be due to mutations. And when you look at the table over here given in the question, C, D, and E have mutations other than those involved in codon in position 526, and there are a number of them in all of these C, D, and E strains. And that means changes in codon occur, and the change in codon leads to a different amino acid being present in the polypeptide, and due to these new amino acids, new bonds form that hold the tertiary structure of the polypeptide, and due to these new bonds, the shape entirely of the polypeptide changes, which in turn means that the shape of the enzyme changes as well. Now, some of these other mutations involved in C, D, and E might cause greater changes to the shape of beta subunit, such that affects the area where rifampicin was supposed to bind, and others might not cause such great of a change, like D and E. Despite having equal numbers of other mutations, D has greater resistance to rifampicin. That could mean that the mutation which is involved in a D causes greater change to the shape, to the side of the enzyme where rifampicin was supposed to bind, and is now no longer able to do so, while mutations in E and C do not cause such a great change. Figure 3.1 is a photomicrograph of a section through lung tissue. So you have some labels over here and also the magnification given. A, state the feature visible in figure 3.1 that identifies the structure in the center of the image as bronchus, and list other visible features that help to confirm this identification. So the structures which you can see over here, all of these lines which can be observed are irregular plates of rings which are only present in bronchus. And you're supposed to mention two other features which are present in bronchus. So alveoli does not have any layers of tissue, but as you can see over here, this wall has multiple layers of tissue, so that could be one of them. It also has a large lumen, unlike alveoli. So those would be the two features which I'll mention over here. B, identify the structure labeled J in figure 3.1. State the evidence visible in figure 3.1 that supports your answer. Okay, so around bronchus, you'd have alveoli and blood vessels in photomicrographs, and since these are the alveoli over here, that means the structure J has to be a blood vessel, and I'd say it is a vein because it has an irregular lumen, an irregular shape. See, the ciliated epithelium labeled in figure 3.1 consists of goblet cells and ciliated epithelial cells. Outline how goblet cells and cilia work together to maintain healthy lung tissue. So goblet cells secrete mucus into the airways, and that mucus traps any pathogen or dust that might be breathed in, and the mucus is then swept away from the gas exchange surface area by ciliated epithelial cells to prevent any infections. 4. In the immune system, a plasma cell develops from an activated B lymphocyte. Mature plasma cells synthesize and secrete antibody molecules. A figure 4.1 is a diagram of a transmission electron micrograph of a plasma cell. The plasma cell can be seen in greater detail using an electron micrograph compared with using a light micrograph. One describes the extra detail of the nucleus that can be seen using an electron microscope. So the details which are visible in this structure because it's seen using an electron microscope compared which would not be visible using light microscope. So you won't see the nuclear envelope as containing two separate membranes. You wouldn't see the pores over here, either the nuclear pores or ribosomes attached to the outer membrane of the nuclear envelope. To explain why cell structures such as ribosomes and the rough and smooth endoplasmic reticulum cannot be seen using light microscope, because light microscope has greatest resolution of 200 nanometers, so it cannot distinguish between 2 points which are smaller than 200 nanometers. And since all of these 3 structures are smaller than that, they are not visible using a light microscope. B. The transition from the activated B lymphocyte to the fully mature plasma cell requires a number of mitotic cell cycles to occur. This process, which is known as clonal expansion, results in a large number of genetically identical plasma cells, which would be able to produce identical antibody molecules. Figure 4.2 describes events A to F that occur during the mitotic cell cycle of B lymphocyte. So you have these events which are not in order. Table 4.1 lists these stages occurring during one cycle of B lymphocyte. The stages are not in correct order, and you're supposed to match the letter with the stage that occurs. Complete table 4.1 by writing the letter of the event described in figure 4.2 that correctly matches the stage of the cell cycle listed. Leave a blank space if there is no matching description for the stage in the list. Use the letter once only. One of the letters in table 4.1 has already been added for you. G2 phase. A centrioles replicate during G2 phase. In cytokinesis, the cell divides into two, which does not have any description either, so we'll leave that blank. Prophase, this has a description in here as condensation of chromosomes. S phase, so the stage for DNA replication, and that involves the activity of DNA polymerase, which catalyzes formation of phosphodiester bonds. And a phase where chromosomes are pulled to opposite poles and centromeres are pulled along with chromatids as well, so that would be E. G1 phase, so the growth phase before DNA replication, and that does not have any description over here either. In telophase, nuclear envelope resembles around each set of daughter chromosomes. C. Clonal expansion also results in the production of memory B lymphocytes. Explain the importance of clonal expansion and the production of memory B lymphocytes in providing protection for a person against an infectious disease. As a result of clonal expansion, a large number of plasma cells are produced, and since these are genetically identical, that would mean they produce the required antibody, which is specific to the antigen that has entered the body and infected, so they would be able to produce antibodies in a large quantity to overcome infection. And if memory cells are produced, that would mean during any secondary infection, a greater number of antibodies would be produced. That would mean before the pathogen causes infection, they would be killed, and as a result of that, the person would not face any symptoms. Myasthenia gravis is an example of a disease where the immune system fails to distinguish between self and non-self. Explain what is meant by this statement. The immune system distinguishes between self and non-self antigens when it's supposed to carry out an immune response, meaning in this autoimmune disease, the immune system carries out immune response against antigens that are self, because they are identified as non-self and not self. And as a result of that, receptors on muscle fibers are blocked by antibodies produced by B cells, which are activated by T cells, which are specific to receptors on muscle fibers. And when those muscle fibers are blocked by antibodies produced, that would mean they are unable to receive impulses which would have been sent by motor neurons, and as a result of that, the muscles stop working and eventually break down. 5. Sucrose phosphorylase is an enzyme found in some species of bacteria. One function of this enzyme is for the production of compounds that help to protect the cell from harmful osmotic changes in the external environment. Figure 5.1 shows the reversible reaction that takes place within the bacterial cell. So we have a sucrose that is broken down to alpha glucose phosphate and a reducing sugar, which you are supposed to name. Sucrose is made up of glucose and fructose. That means X has to be fructose since the glucose is already present in the other product. B. In the absence of sucrose phosphorylase as a catalyst, the reaction shown in figure 5.1 would take too long to occur to allow the bacterial cell to function efficiently. Explain why the reaction shown in figure 5.1 proceeds at a much faster rate in the presence of enzyme. So you're supposed to explain why enzymes speed up a reaction. They do that by lowering the activation energy of any chemical reaction and they put strain on bonds. An enzyme that catalyzes a reaction of commercial interest needs to be investigated to see if it is suitable for use in industry. For example, immobilized enzymes may be used as they have a longer shelf life than the enzyme free in solution. Many industrial reactions are carried out at higher temperatures to minimize contamination of products by microorganisms. Figure 5.2 shows the results of an investigation to compare activity of sucrose phosphorylase free in solution with immobilized sucrose phosphorylase, which is an immobilized enzyme, at different pHs. Figure 5.3 shows activity of free enzyme and immobilized enzyme at different temperatures. So here we have varying pH and activity of the two enzymes. And figure 5.3 shows activity changing with temperature. Alright, with reference to the results shown in figure 5.2 and 5.3, discuss which sucrose phosphorylase enzyme, free or immobilized, is better for use in industrial reactions. Let's go back to the figure 5.2. Here we can see that the cross represents immobilized enzyme. The percentage activity of immobilized enzyme is greater than the activity of free enzyme for the most part. So that makes it a better option. Also, in temperature, we can see that immobilized enzyme has a higher optimum temperature, which makes it more suitable since, as the question mentioned it, many industrial reactions are carried out at higher temperature to minimize contamination of products by microorganisms. So it would make a better option. You can also mention the reason it has higher activity at a higher temperature. It has great activity at higher temperature That could be due to its thermostability or the fact that its hydrogen bonds are not as much affected at a higher temperature as those in free enzymes, because those bonds would be holding the shape of the enzymes, and if those are broken down, the shape of enzyme would be lost and its activity would start to decline. 6a figure 6.1 shows an oxygen dissociation curve for adult human hemoglobin. An increase in partial pressure of carbon dioxide in respiring tissue causes the Bohr effect, which would shift the oxygen dissociation curve to the right of this curve in the question. One sketch on figure 6.1 to show how the Bohr effect changes the oxygen dissociation curve. Right. So the shape would remain the same for the most part, except it would shift to the right. This basically represents hemoglobin, where present in areas of high partial pressure of carbon dioxide has lower affinity for oxygen, or in the same partial pressure of oxygen has lower percentage saturation. To explain how an increase in partial pressure of carbon dioxide produces the Bohr effect and state the benefit of the effect of this effect for the tissue. So you're supposed to explain why increase in carbon dioxide causes a decrease in affinity for oxygen of hemoglobin. When carbon dioxide, the partial pressure of carbon dioxide is highest in respiring tissues, so carbon dioxide moves out of the cells and into the capillaries that are carrying red blood cells, and in the cytoplasm of red blood cells it reacts with water to form carbonic acid, and then it dissociates to form hydrogen ions, which then react with hemoglobin or displace oxygen from hemoglobin to form hemoglobinic acid. This not only increases the pH of blood, therefore decreasing in the acidity of blood, but it also frees oxygen from hemoglobin, which is then supplied to the respiring tissue. So you need carbon dioxide in, so that is the use of carbon dioxide in respiring tissues, so that they can gain oxygen, which is bonded to hemoglobin molecules. In your answer, you should state terms that you'd use in comparison, because carbon dioxide is present in all cells, but in respiring cells, where partial pressure of carbon dioxide is the highest, the effects that we're supposed to describe in this answer are going to be the greatest. The carbon dioxide is transported across the cell surface membrane of red blood cells using a different mechanism to the transport of hydrogen carbonate ions. Name the different mechanisms of transport used for carbon dioxide and hydrogen carbonate, and explain why they are transported across membrane by different mechanisms. Obviously, carbon dioxide is not a polar molecule, so it is soluble in the hydrophobic core of the membrane, whereas hydrogen carbonate ions are polar molecules, so they would require an aqueous channel provided by transport proteins in the cell surface membrane. So carbon dioxide moves by simple diffusion, whereas the ions move by facilitated diffusion. Alright, we are done with this paper. Thank you for watching.